really fun evening. But before that, what is Genspace? Genspace is the world's first community biology lab, which is my favorite sentence to say. Get to say that like once a week. And what that means is it's a new model for open access to science. So it means that doing research and thinking about biology, thinking about the systems that run our world can be something that is rooted in real lives and real communities and connected to the things that you care about. And so that's what a community biology lab is. We're founded in 2009 in a living room in Park Slope. Literally, our founders got together and they were like, biotechnology, gene editing technologies are things that affect our everyday lives, and they should access to the tools that drive those technologies should be accessible. And so they started doing basic bacterial transformations in a living room in Park Slope. And that is a picture of what that looks like, which you can kind of see because it's a little sunny. And so our mission is to foster a safe and inclusive space where anyone, regardless of background, but particularly for people who may not have had a chance to get engaged with science yet, can come and do science with their own hands and drive forward in a way that is connected to real life. And so we envision a world in which everyone is empowered to use the life sciences to develop applications and explore questions that are rooted in their real lives and communities. So you are part of that here tonight. Team. Can I get the staff to stand up, please? Can I have clap for the staff? We have an incredible team that run this space, and after the talks tonight, if you want to talk to them about getting more involved in classes or membership or some of our programs, please seek them out and talk to them about those things. We have a couple of buckets of programming at Genspace. So we offer youth programs, we do events and outreach, so we'll do public programs here, like the one you are currently attending, but we also go out in communities all over the city and set up tables to extract DNA from strawberries. We make objects out of bioplastic, anything that lets you walk away having learned something that you can take home and tell people about having a meaningful experience with science. We classes and workshops, so you'll hear a little bit more about what's coming up at the lab after the talks tonight. One of our signature courses is called Biohacker Bootcamp, where you can sequence your own DNA, you can make bacteria change colors, you can go from having experience with science to basically doing molecular biology yourself in a weekend. So pretty cool if you ask me. We also have a lab membership program. And so this is where you get to mutual learning group doing projects or work on your own science projects. We also engage companies, so these cool pipetting robots over here. This is a lab automation company called OpenTrons that was founded at Genspace in 2012. And so we offer from education all the way up through commercialization. Okay, so I promised that you would get to hear more about our FIT scholars. And so from our classes and workshop to our membership program, there's perhaps no program that Genspace offers that is more exemplary of the pathway from education to independent research that can take place here than our partnership with the Fashion Institute of Technology. We've been running this program in 2017, and it's a semester-long program where we bring undergraduate students from FIT to take classes at Genspace and engage in mentored independent research. And so they explore questions at the intersection of biology and sustainability and come up with processes or applications that build a more sustainable future. So you're going to hear about their projects tonight. And in order to introduce the program, I'm really excited to welcome Dr. Karen Pearson to say a few remarks. I just have to read Karen's bio because it's really impressive. So at FIT, Dr. Pearson is the Chair of Sustainability of Science and Math. She is a chemist by training, and she has numerous accolades and recognition as one of the top most 100 influential women in STEM. Wow. Yeah! And so partnering with is a real pleasure every spring. It is through her thought partnership and guidance that we're able to make the program what it is, bringing students who are working in the fashion industry and have interest in business, in marketing, in design, and give them the tools of the life sciences to realize something really incredible at the intersection of things. So thank you, Dr. Pearson.
Well, first of all, thank you. I first want to thank the amazing team here at Genspace. The Genspace team really allows us to offer this program and to help it be a place where our students have this opportunity to really offer a solution to the biggest global challenges. And that, as part of the State University of New York and part of the Fashion Institute of Technology, we are so excited by. So first of all, thank you to the entire team. But it's not just the team. So many of you sitting here in this room have offered mentorship or been an educator in one of the classes that our students have come and taken, not just the students who you'll hear from tonight, but we actually have a number of students who have the opportunity to come and experience the national space of biotechnology here at Genspace. And I really want to thank all of you. I don't know you all by name, so I'm sorry, but you're all amazing, and you've been amazing, not just this year, but over many years. So thank you all. So really, again, we're so excited to have this program that has been brought to us and been able to grow with partnership across the college and with partners in the foundation and the entire space of our partners who really have embraced this. And FIT industry partnerships are key to our students' success and how they move forward, and we're so excited that those same industry partners who are part of what's considered to be the fashion industries are also partners here and help support this program and numerous other programs that allow us to support and engage with your entire community. So thank you.
Without further ado, we are going to have some of our FIT scholars present. Our first presenter is Olivia. Olivia came to us wanting to know how she could support her pets and her wonderful cat. And so I think that there's a lot of areas in which you will see some overlap tonight. There's a big materials focus, and so trying to make materials sustainable and also not as harsh for our bodies and the bodies of our beloved pets as well. And so Olivia, I would love for you to take it away about telling us about your pet Band-Aids.
Hi, I'm Olivia from FIT. My major is fashion business management, and my minor is journalism and sustainable materials and technology. And I'm now became the senior, and I'm also the vice president and the student government association at FIT. So this is a background, the current problem. Many pets experience allergic reaction or skin irritation due to conventional adhesive bandage. These reactions can include redness, itching, or swelling. Most commercial products also contain synthetic chemicals or materials that aren't ideal for animal use. This leads to me ask, can we create an alternative that is both safe and eco-friendly? So these are my research questions. While eating fruit, I noticed how the juice sticks to my face and hair. When I tried wiping out with tissue, the paper cling to my skin and tear it apart. That everyday moment made me think, if fruit juice is this sticky, could it work as a natural adhesive? This simple question became the starting point of my experiment. So I choose 8 of fruit to take this experiment: banana, mango, apple, tangerine, aloe vera, and some aloe vera powder and lemon. Following the research, are all safe to pet and human skin. However, lemon and aloe vera cannot be used in final formulation because lemon is too acidic and it can cause stomach upset and skin irritation. Also, aloe vera contains compounds that may toxic to dogs and cats, so they were just used to comparison controls, not as viable ingredients. So I want you to use natural preservatives because my ingredients are from the natural thing like fruit. They needed more bacterial things. So I use honey, which is widely used in both human and veterinary care due to its low pH. Natural hydrogen parasite release, high sugar content, and it is also safe for external use on animals. And I also use grapefruit seed extract. It extracted from grapefruit pulp and seeds, and it demonstrated to be effective against. And it also used in pet product and also human skincare product too. So these are control pH tests and result. First control was current market pet Mayday spray. It pH was 5.4, and second one is cornstarch and water. I boiled them. Its pH was 6.0 to 4.9 because I add honey and GIC. Third one was cornstarch and pectin, which was below the 0.01 and 0.0. So conclusion was adhesives with pH below 4 may irritate animal skin. So finding natural adhesive with neutral pH level was really key for my experiment. So these are my general process. First, all fruits were sterilized externally before use to prevent contamination during extraction. Second, the chopped fruit with peel and its juice was placed into a pot along with 55 milliliters of water. The boiled mixture was poured through a strainer to remove solids. Additional liquid was extracted by pressing the solids through the strainer to capture remaining juice. And then I collected liquid was returned to the pot and combined with starch half or 3/4 teaspoons. I also split by 3 from the finished adhesive. First one is control with no additive and no changes. Second was I added honey, and third one was added some grapefruit seed extract, and I also under pH level with digital pH testers. Those are the result of the fruit-based adhesives. Those 8 fruit have each 3 result, and you can see it there too. And this is one of general process. I also did application test. I pressed it with medical gauze to the fox fur the fabric, and I just want you to test like is it far apart or it attached well. So this is fruit adhesive pH test and result. As you can see, banana, mango, aloe vera showed neutral pH level, which is ideal for further testing. Highly acidic fruits like lemon were excluded due to potential skin irritation. I researched that dog and cat skin is more than human skin with a pH range of 5.5 to 7.2. Only banana, mango, and aloe vera were tested in all 3 conditions with base, honey, and GICE on LB plates. And for comparison, I used apple, lemon, and aloe vera powder with only fruit base. This is test on LB plates to see if there is any change in microbial contamination or antimicrobial properties. But since today I couldn't see any of changes, I also tested in 30° and 30°. This is future lab steps. I just want you to test antimicrobial properties. First, determine a commonly found on pet skin, and then plate in gauze patch or adhesive and measure in inhibition zone. The downstream step is IACUC approval, veterinary consultation, toxicity testing, microbial safety, and shelf stability tests, regularity classification and labeling, and pilot testing in shelters or with pet owners. And going further, I believe collecting veterinary schools and materials researchers can open new pathways in animal care. Veterinarians know the biological and clinical needs, while researchers can develop safe, tailored materials to meet them. My project also taught rising interest in homeopathic or plant-based solutions. While they may not fully replace conventional treatments, they can serve as supportive care options. Currently, most pet wound treatments involve oral antibiotics. Having effective topical options like neutral adhesives with antimicrobial properties would allow for easier, more localized treatment. This could be especially helpful for minor wounds in reducing the needs for systemation. So these are future design application. I just want to launch to send just in case if the pet has mango or the banana allergy. I just wanted to use eco-friendly packaging too, and I want veterinary partnership too. And this is the face adhesive recipe. If you guys want to do it in your home, maybe you can just try.
Awesome.
Thank you.
Yes, yes. Do we have any questions for Olivia? Yes, Jeremy.
So what gives the adhesive product? The sugar and the...
Yeah, because fruit has richer pectin and sugar and fiber. They have some sticky contents, and I also added the cornstarch and added to boil them, so they make more sticky and some glue type of thing. Thank you.
So, did you use this on your pets and did you lick it off?
No, I just tried on the fox fur fabric. Maybe you can see on there. So I just put on the medical gauze first and then pressed on the fur fabric. So just test they are good or not. So first I tested in 15 minutes, and some of them were not good at glue, but after like 1 day or 2 days, they just stick well.
Does the glue stay ever get solid, or does it stay a semi-liquid?
I'm sorry.
Does the glue ever get solid, or does it stay a semi-liquid?
Solid? Does it turn hard or does it stay soft?
Oh, actually, it is. It stays soft, but like maybe like 12 hours later, it is more stick well. Yeah, it is more solid.
This is more so for my clarification, but in terms of application, is it something that already has the adhesive applied to it, or is it a situation similar to what you described where you have like glue gauze and then you add the glue to it and that's like a separate fluid on its own?
Can you say again, please? I'm sorry. I can.
Oh, I said, is it a situation where it's like a Band-Aid, for example, where the adhesive is on the actual, or is it a situation where you would use like a gauze
Oh.
and then it's a separate fluid?
So for first, because I only did as a glue thing, so as I showed in product, I just want to release as a glue type me first, and then after, if it can stay more thin way, maybe we can just release the product. But first, I just want to make a glue type. So maybe you can just buy some medical gauze, and then you can just put with that glue.
Do you see this being used for veterinarians, such as post-surgical intervention, or do you see this kind of being used for somebody who has a cat that got a scrape and want a quick solution to kind of heal the cat wound or dog wound?
To be honest, I want to use really wide range, but first, because it is just first step, I just want to suggest that use really minor ones first, because right now I couldn't find any of microvirus change right now, but like we need to more experiment and test, so I just want to suggest a little minor ones first.
What's the shelf life? Have you tested anything like that?
I'm sorry?
Shelf life?
How long does it last?
In right now, I think they really stick well now, because I think it is maybe 1 week, I guess. They stick well, but when I try to take away with water, it did out well. So I just suggest maybe some pet owner want to take off them, I can suggest with water, removing will be really good.
Thank you. Thank you, everybody. So, our next fabulous scholar is actually on her way to South Africa right now. She is doing a study abroad program over there for the next, but she did a wonderful project here in isolating proteins from invasive jellyfish. The idea being to create hydrogels that can be used in agricultural applications. She worked with Katie, who has wonderfully arrived, isn't there in the back. So if you have any questions about the whole process, feel free to ask Katie as well. But we did record a little video beforehand, but the light is not helping us right now. Okay, so, maybe it's a little better. Okay. And let's see how the sound works.
My name is Georgia. I am currently in my junior year at FIT. I am studying textile management. My name is Katie. I am a year removed from graduating with my PhD in biomedical engineering, and my scientific background is largely in synthetic materials as well as cellular biomechanics.
Awesome. So, Georgia, can you tell us a little bit about your project and why you decided to focus on this jellyfish in particular?
So my project here at Genspace was focused on actually creating a hydrogel for agricultural applications using collagen from jellyfish. One of the reasons we would use collagen from jellyfish is because that would create a hydrogel that would be hopefully biodegradable. Also because jellyfish are an invasive species in large parts of the world, and it's kind of this excess opportunity.
Katie, can you tell us a little bit about what hydrogels are and what they are used for?
Absolutely. So a hydrogel is basically a soft biomaterial comprised of a hydrophilic or water-acting polymer that has a strong propensity for retaining water. And their applications are pretty wide range. They're almost limitless, honestly. Most of the ones that I'm familiar with are hydrogels for medicinal applications, such as plant-based hydrogels for spinal disc repair or injectable soft tissue repair. In my graduate school career, I also spent time using more synthetic hydrogels for experimental modeling of cellular biomechanics in various different disease states. But over the past 2 months now, it's been really exciting to learn more about their applications outside of use in cellular experimentation and medicinal applications, both in environmental sustainability and agriculture. I've been helping Georgia with her project and reading a lot more about this realm of materials that I really had not been experienced before.
Can you tell us about how you would want to apply the hydrogels that you would make down the line?
So mainly they would be used in agriculture. I think my initial idea was using it kind of as a way to use less of our drinking water when farming. It's about 40% of our clean water goes to our agriculture, and if we can introduce something like a hydrogel that would allow us to limit that, but still kind of benefit the growth of, especially when it comes to things like food. I think also in terms of just growing up in New York City and urban farming, a lot of our soil, might be high in iron, or it's depleted a lot of its nutrients. So creating something that we can put into soil that would give it structure and kind of these mechanical benefits, but also maybe possibly eventually slowly introducing nutrients back into that soil to benefit the growth, to make farming more accessible for everyone.
Awesome. So you guys have spent quite a bit of time together in the lab. It's been amazing to see. What kinds of lab techniques did you use? And Katie, what role did you play in teaching some of those lab techniques?
Sure. So we had a list. We had to sit down and go, what have we actually done? So we had pipetting was a big one, learning about the first and second stop on a pipette, which is something some grad students didn't really understand when I worked with them. Molar measurements, creating solutions, pH measurement and solution pH adjustment. What else have we done? Dialysis, that was very exciting. Experimental design, and kind of learning to really what Katie helped with also is kind of translating research papers. There's a lot that happens when you're reading these kind of documents that you're they're using very technical terms, and it's either you're Googling every other word, but with Katie here, she was really able to just guide me and really mentor me. She's the best. But what else? Also piecing together parts of experiments and kind of reading through a paper and finding what we need, what we don't, what we can expedite given our time frame. Yeah, that was a big thing. I think that's what my largest role has been translating some of these more complex academic papers, trying to piece them together because some of these protocols, because not enough people are talking about jellyfish or using them. Not enough people are talking about jellyfish. They're not talking about them, using them experimentally. Some of these protocols are, quite frankly, hard to follow because they're not giving us precise measurements or time frames for a lot of these protocols, and also given our own time constraints and just limitations of the and just the materials that we have on hand, trying to figure out a way to adapt these protocols to be able to get her product done to the closest approximation we can in this, what was it, 2 months you had? Oh my gosh. Yeah.
And what did you learn about protein extraction, Georgie?
Well, I learned what protein extraction was. I think I haven't had really any lab experience, and I think a lot of what I was coming into is just what I've read. When I first thought of protein extraction, I thought extracting the protein, with a needle maybe. I didn't know how it worked, but really what it is it's kind of you're removing everything else. And kind of like you're looking at something, you're looking at your source material, which really can be anything. For us, we use jellyfish, and the fastest way to get it was kind of shopping around, and so I went to a couple fish stores and tried to find these jellyfish, but it can really just come from, protein can come from anything, and science really is accessible.
All right, Georgia, so you have a few more months with us. We would love to hear a little bit about how you're going to expand this research in the future.
So the next steps would be applying it and creating a hydrogel from that. We would then be testing its mechanical strength properties, seeing how does the jellyfish collagen impact those hydrogels. We want to see when it decomposes, what does it leave in the soil, how long does that take. We want to see the structural benefits it has to the soil, as well as just how long it takes to release water and then maybe reabsorb water from rainfall. We'd also maybe want to see if we can add nutrients to give it back to the soil, kind of just testing further.
Awesome. And Katie, just one more quick question. When you have a protein, how do you turn it into a hydrogel?
That is a complicated question, actually. But I know for the large part that the creation of hydrogels, most of my experience has been with chemical cross-linking. But I think that when you're making a hydrogel out of just, like if you were trying to make a just collagen-focused protein, you could do maybe more of a mechanical cross-linking process. But basically the long and the short of it is you need to pick if you want to have just a protein-based gel, or if you want to have some sort of base alongside that protein, whether it be a PEG, or in the case we're thinking about sodium alginate, and then how would that impact the final results of the experiments that Georgia wants to build upon, and then how do you then cross-link those two materials to make them stay together long-term, that way they're not just in a floating solution and they don't want to bind to one another, so.
And I have one more question. How would you describe your experience at Genspace over this semester, Georgia?
Honestly, it's been amazing. I think it's just been a fun place to come to, and I think I've been able to meet so many people from just, there's not just one type of person that comes here, so I think it's pretty amazing. Like, there's people from all sorts of fields at different stages in their career and their education, and everybody's coming here to learn. I think, honestly, like, this has really just given me an opportunity to learn also more about myself and what I want. And kind of, thinking about, I'm in my junior year of college, like, where do I want to go after this? What do I want to explore? Kind of, like, figuring more about myself, too, through this, like, learning environment. And it's just been amazing.
Awesome. Thank you guys so much, especially for all your hard work. It's been a joy to have you guys here. Thank you.
If you have any questions after we are done with our presentations, I know, Katie, you'd be willing to perhaps answer some of those, but Georgia has been such a member of the lab, and we can't wait to see the hydrogels that she comes and makes from there. Yes. That was Georgia. So, all right. From there, we're going to move on to our last FIT scholar, Emma. Has taught me more about my bath routine than I ever thought possible. I am a user of bath bombs, and I love them dearly. But Emma alerted me that there's some things I should be kind when using bath bombs, and perhaps I shouldn't be sitting in a bunch of colorful, glittery water for too long. Can't imagine why. So Emma is here to tell us all about the chemistry of bath bombs. Able to see some of them that she has made. They look very nice over afterwards as well. But there's also some other sort of a patient advocacy work as well that Emma's focusing on about sort of where that bath bomb project sort of led. So, please take it away, Emma.
Well, that was a really good introduction. Said a lot. So I called my thing FOM, thinking about how to make pH neutral bath bombs. And I don't know if you guys have, like, what spurred my interest in this was seeing studies about how bath bombs weren't, like, healthy for vaginas in particular. Not sure about penises, I'm sorry, guys, for everyone who knows that. I'm not researching that. But this is just our final test. So pretty much in my personal fine arts, probably focus on how to create education or educational resources through visuals, and personally I really focus on women's rights, anyone who has a vagina, whatever you fall under that range of woman or whatever, and like the rights and how you exist within that body. And so I really wanted to take that and put that into my time at Genspace and find a way to make something scientific out of it. And yeah, I think we can go on to the next slide probably at this point. 1 second, guys, sorry. Okay, so that kind of starts into my research. So on the left here is just like the range of pH. So on average relative, the environment of a vagina is like 4 to 5, and so it's pretty acidic, and that helps keep infection down, help prevent UTIs, and all sorts of infections like that. And the one that really interests me is this graph on the right here, which tested different parts of the like cervix, the vagina, like the outside of it, all the way up into like the uterus, all of that, and tested in the range that existed. And as you can see, like the top there is 8 and the bottom there is 3, so there's a huge range, and each one of those graphs represents someone who's in early term pregnancy, late term pregnancy, and then someone who's not pregnant. What I noticed a lot is that a lot of research I was finding was related to like women and fertility, and so it was really hard to find research that was like just about women's health or just existing, and it had to be related to like pregnancy or something somehow. So that was like piqued my interest, I guess, because I really am passionate about, you know, I don't know, having rights to your own body, as many people are. But so I just really wanted to, like, that really piqued my interest, and I really wanted to like think about the advocate of it and kind of think about maybe, because the more I was researching, the more I was realizing how little I actually knew about my own body, and that was really surprising and disturbing even. And so I really wanted to create some sort of like fun that could help other people learn. And so here are 3 main bacteria that live in a vagina. I made a little sign. You can pass it around if you would like. Yeah, so if it gets to you, you'll learn a little bit about them. And then going back to the patient advocacy part, yeah. So about 20% of women report experiencing mistreatment during maternity care, and then there's higher rates for women of color, percent higher. And for me, that was really shocking as well to see that statistic, because that's like a field that is arguably made for like the vagina, but it's not serving like the people who are actually going in. So that was like, you know, upsetting. I wanted this project to connect the scientists who have the ability to start genuinely researching these things, and then also to connect to the patients and the people who need to have these resources. And then yeah, that's a little guy that's being passed around. So my research question when you're getting into it, thinking about the bath bomb part, was really like, how do we create a bath bomb that has a minimal effect on the pH level of vaginas? And honestly, I had very little faith in my project. I wasn't sure because, you know, you're thinking about a base acid, like neutralization, yeah, process. So it's like you really need to have a strong acid and strong base, and then it comes together as, you know, neutral. So I wasn't sure it was going to turn out well, but we'll get into it. So I needed to establish 2 main procedures. One was to kind of test the materials, and then the other one was kind of like how to create like the production process of a bath bomb, and like keeping that I guess effective, and like what it actually takes to like produce something. And that kind of had a lot more problems than I thought, or a lot more things that came up than I thought. And then for my variables, again, we're testing for the independent what materials are being used to create bath bombs and the concentration of each material in the recipe. So that's like the acid and the base concentration. And then the dependent is the pH level that we're testing that. And then some controls that we had were just water to kind of have the no reaction kind of comparison. And then positive controls included using a traditional bath bomb as a comparative study, and I'll keep with it. And so, so for experiment 1, we're controls here. So this is just like the pH strips and then the little measuring setup. And then the comparative study, the main thing that came out of this was that it was like the water was neutral after the reaction. So it kind of made me like, why is it then affecting like pH? One would think like, you know, neutral would do nothing, but it kind of isn't. The way that kind of set up, and that kind of led me to doing more research about just what makes up like vagina, and that's where I learned that it's really acidic and that it's like temperamental, so even just having something neutral would really like offset that. Experiment 2, it was more of like a production type of kind of like figuring out how to like produce things. So while it wasn't really intended to be figuring out how to like produce bath bombs, and that wasn't really like the point of it, it kind of became like, this was like a degradation test to see how well they would like hold up with time. As you can see, some of them did not do well. And then I also tested pH levels here, and in that experiment I used a consistent base to test against the acids, and then I chose it based off of these results. And then in experiment 3, I tested those two acids as a control against the base to find whichever one would be the most neutral. So in conclusion, I guess my hypothesis was right. The commercial bath bombs are kind of doing exactly what I kind of created in the lab, as they both were pretty neutral. But nonetheless, the idea of like patient advocacy kind of came up through all of this, and I really wanted to focus on that. So also, yeah, what I would do differently, I think the human aspect of my experiment was really like left out of it. Like this is a, testing to see what the effects are on people. And obviously here at Genspace we can't do that, which is okay. I think if I were to continue this, I would really want to be able to test like with people. And I really learned how to pivot when your experiment isn't going how things are planned. Like I guess expected it as planned, but kind of how to what do you do when things aren't working out? And so this is what I decided to do when things weren't working out. I made this little website just to show this is how I feel like should display themselves. I had these those would be designs you could download for free, and then you could share them wherever, your local coffee shop, your doctor's office, wherever. So I just think there's a certain responsibility. I think businesses that sell to people, that sell to people that would really affect their daily life. There's a certain standard that some of them aren't always living up to. This was a really cute idea I had, or not cute, but where there could be a little form on the website where you could connect with doctors and just have general questions answered before you went into doctors' offices and stuff, so you could know what the right question is to ask. And then you'll see these on the little boxes I have, but I made different little hands in the little box. Yeah.
Questions for Emma?
I saw the recipe was listed as 1 part acid, 2 parts base. Do you think shifting the ratios would let the end product be more acidic, or acidify the water in general, and hopefully match, I guess, the natural pH, and hopefully that could have an effect?
Yeah, definitely it would. Changing the ratios would make it more basic. The problem I kind of found with that is that your skin has a different pH. It's about 5 or 6 if I remember correctly, so it's still on the acidic side, because I don't know, I learned a lot about your body is really acidic actually to fight off all the infections. I didn't know. So that would cause a little bit of problems because you don't want to be bathing in acidic water. So it's kind of a hard balance that I found out was a really hard balance to create.
Remember the body has a pretty high acidity in order to fend off infections. This is kind of separate from your research, but do you know if there's any connections between the acidity of different parts of your body and your acidity?
I have no idea, but that's a good question.
Really cool project. I've never used a bath bomb before, and they look very interesting. But I was curious, in your research, did you ever find the composition of the bath bomb had any microplastics in it? And I remember in the beginning of the presentation that some of them are glittery and some of them are kind of varying in color. Have you done any research, or have you come across any studies where the exposure to microplastics in relation to the water you're sitting in affects your performance?
That's also a really good question. It didn't come up for me because I wasn't using it. And the commercial bath bombs I had didn't really have those. But that would definitely be something that should be researched.
Have you thought about also effects on the microbiome?
The
effects of the bath bomb, if they have some detergents or something that affect the
Sorry, one more time?
They could affect the microbiome.
Like,
It could also affect the general health.
oh yeah. That was like a big. I kind of skipped right over that part. But yeah, so would definitely that is like the thing that is being affected when the pH gets dysregulated because of the bath bombs. It's because the bacteria is getting killed off or something. Like there's some sort of like disturbance in that balance because that is what really is the pH. Yeah.
Awesome. So, we have had a wonderful suite of mentors that have helped us in this FIT program, and so you've seen a little bit of a slice of the wonderful work that we do together. Katie is here. We also had 2 other mentors as well, Aiko Yamada and also Taja Ellis. We want to thank them as well for all of their hard work working with our scholars. Here at Genspace, as you've heard, you have a variety of different ways you can get engaged, and a scientist can come from everywhere, to quote Ratatouille. And so I would love to now sort of fold into how you can become sort of a scientist and how all of these processes sort of start from a young age, as from a high school student to becoming a career life artist doing fabulous things. And so next portion of our presentation, we're going to be hearing from a former participant in BioRocket. He was there in 2021. It is our research internship program that we run for high school students over the summer, well, spring and summer. And then he took all of those wonderful researching skills and applied them to a study abroad program. And so we'll be hearing from Bashir next, and then from there we're going to be hearing from 2 of our members here at the lab. So, Bashir?
So, hi everyone, my name is Bashir Bajaha. I'm currently I'm going into my senior year at the City College of New York. And currently I study anthropology and biology, and I'm looking to do something maybe in animal behavior. And so this past January, in the winter, I did a study program for marine biology in Costa Rica. It wasn't my first marine biology experience, but it was my first experience doing a study abroad out of the country. I do want to mention that it was made possible due to the Gilman program, which is basically a scholarship that helps fund you to actually do the study abroad program. And so I'm doing this presentation also on behalf of them as well. So I just want to introduce Costa Rica and also my time at Genspace. It was in 2021, so it was I also, so for my study abroad, my Genspace project that I did, it was on biofilms with microplastics, and also how we can possibly use microplastics to be able to remediate in a clean way instead of using toxic metals that can actually harm the environment, especially in the Hudson River. And so Costa Rica from a glimpse, I remember before going, people were saying how the people are, there's a term that they use, pura vida, which means basically pure life. And so a lot of the people are really laid back. They also tell you that if someone says, like, yes, they'll do something, it doesn't mean that they're gonna do it like right away. They take care of things. And so I also got to experience this firsthand because the way the program was structured is that we stayed with host families. And so I stayed with an old lady and her husband, and through time to time, she'll in-laws come with also her daughter and their spouses as well. And they also had a dog too, so that was cool. And so the way that everything was structured is that we would have dinner and everything with them, day and night, breakfast, lunch, and dinner when we could, because the way the program was also structured is that we did do a lot of excursions, which were like road trips, and we went to a lot of different places in the country. So when we actually did get to stay at home, it was fun to live with family, like to actually feel like you're in a family in a country. And so the first excursion trip that we did was to the North Pacific, which is basically on the western side of the country. And so we visited the intertidal zone, which had a lot of mussels, barnacles, and sea cucumbers. And there's a few pictures here that we actually saw a dead porcupine that probably got trapped when the tide actually like moved back the day. And so we did a lot of snorkeling, which we used the ID to be able to identify the fish that we saw. There's a lot of pictures that I wanted to include, but they were taken on like a professional camera, so they were the file size to actually include into the presentation. So that's not good. And also, we went to a mangrove and we saw a lot of fiddler crabs. And they were really cool because they were like tree crabs and they had like the small claw and then the really large claw. And they actually, like when we were on the motorboat, like going under the trees and seeing all the different kind of mangroves, which like the white mangrove, for example, has these branches that they kind of like stem under, but it's like wood, they're not really like leaves. And so they were getting in the way. A lot of tree crabs would be like on the branches, like hanging like right above you. And we also saw black iguanas, we saw green iguanas. And there was also the tour guide said that there were nighthawks, which was also, he was like, really excited to see because they're usually nocturnal. So we saw one that was resting there during the day. But of course it's taken on professional camera, so can't include it. So in the Caribbean, that was on the eastern side of the country. And so this is from the, we were supposed to do like a coral reef restoration, but unfortunately the waves that day were really bad. And I also don't know how to swim, so we couldn't do the reef restoration, but here basically getting taught how to attach prosthetic coral, which is basically to help enforce and like encourage a more coral to grow on top of the actual structure. And so we actually did reef restoration and with lionfish as well. So we met a tour guide a fisherman who hunts lionfish for a living, because the bad thing about lionfish is that they're an invasive species. And so they actually originate from the Indian Ocean, and over the past 20 years or so, a lot of them came into the Caribbean Ocean, actually starting to spread into like Florida too. And so we actually got to eat lionfish. It tasted like regular grilled fish. It was still really good. And so we actually he showed us also how to gut the lionfish, and so how to identify male or female, and how if you catch a female lionfish, she is actually carrying eggs. So that would also be like a bonus catch because you're catching like the babies as well. And so, and of course there's like this is a photo from one of the beaches in the Caribbean, and you can see like the opposite side, I think it was like a tropical rainforest as well. And so next we went to Manuel Antonio, which is it's a national park, and we also went to Caño Island, where we did more snorkeling, and we actually went on a tour into one of the rainforests. And the top picture, it's kind of hard to see, but it's a baby eyelash viper, and so it was like perched onto a vine, and it was really hard to see at first, but the tour guide was really good at spotting them. And so over here, it's a giant American crocodile that we saw it was like a little ravine. It was a big ravine actually. Like, there had to have been maybe like 50 crocodiles in like that area alone. And so we also saw black howler monkeys, and there was a vine snake too, which is also hard to see. And both of them were actually babies, both of the snakes. And a lot more photos that I wanted to include, but I couldn't. And so this was the, I shouldn't put the sharks, I maybe should have separated them, but the shark lab that we did was actually a little bit different, because we did a lab with sharks, but we didn't get to see like live sharks. So for the live sharks, we used like old fin samples, and we were able to extract DNA and to be able to actually like determine how much of the DNA was left over. They were really old, and they were like old and dusted, and to see how much DNA was left on it. And so, but the ones that we did get to see in person, sharks and rays are related. So we got help from a lot of the marine biologists in the area, and they caught a lot of rays, and some of the species that we caught were guitarfish. There's the electric ray and butterfly ray. The butterfly ray is not shown, but here's the guitarfish, and there's the electric ray on the top. And it was fun because we were trying to talk about the electric ray, but unfortunately it didn't do it, so yeah. But for the guitarfish and the other rays, they also taught us how to identify male and female. It's similar to like crabs, how like male crabs have like the right on like the underside of the carapace. And so I just want to also highlight the Gilman program, because I want to give this presentation to also encourage other people to study abroad, especially if you're at a program, or even just you want to do an internship abroad, because they can provide up to $5,000 in funding, and you also get access to the Gilman network, which helps link you to other people who may be studying abroad in your country or who may be just in your. And so, yeah, these are some other reasons to study abroad. You get to have a lot of fun. These are some of the people that I met. This was at the ravine with the crocodiles, and this picture. And you also get to earn credits towards your degree. You get to witness new cultural experiences. And if you receive financial aid, it can help cover the cost if you don't get a scholarship. And yeah, that's basically my presentation.
Do you have any questions for Bashir?
Is there anything that you learned abroad that you're going to bring back to the rest of your college experience?
I think for the most part it has to do with biology because I'm minoring in biology, and so everything that I've done there with the Shark Lab, it kind of like related to what I have done in the past and have been learning in school. And so yeah, I think that's really the main thing. I think between like the two countries, there's also like the huge like cultural shift as well. So that like helps with things as well.
What did you use for presentation?
Thank you.
What did you use for the coral retrofit?
The coral?
Yeah.
You mean like the structure, right?
Yeah.
Oh, it seemed like it looked like it was kind of makeshift, but maybe I think it like steel pipes that were like kind of like structured into like a base that coral could be able to like wrap around and grow on. We also use like those plastic zip ties to be able to like tie them onto it to help with that as well.
What was your favorite animal or
That's a good question. I wanted to see a jaguar, but like it's like really hard to see. But there's also the an agouti, like the small. It looks like a capybara, but the really mini one. We saw a ton of those. And there are also they had like those giant marine, not green iguanas. Yeah, they were like running around a lot too, around the beaches and stuff. So I think those.
So given your, I guess, experiences in Costa Rica, like what sort of like science projects or research projects is literally like on an estuary would you like recommend to biology, community science folks here in New York City?
So your question is like what?
What science or research projects would you think would be interesting to pursue and apply here in New York City based on your experiences in Costa Rica?
That's a good one. So I think at the end of the program we also did like presentations, and I think for my group we did it based on the intertidal zone, and so we could possibly apply obviously the intertidal zone in Costa Rica is a lot different than New York. So and I think there's also like endangered species in New York, like plovers as well. So we could possibly see how we were measuring how the different tides could affect how many species you find in the tide pools, and so we can see how we could apply that to New York environment, and also how to find a way to prevent the endangerment of the plover that I mentioned.
Wonderful. So, our next presenter is Jonah. He's our lab. He came to us as a product designer looking to understand how to add biology into his practice. He had this wonderful idea that he will share with you now. And he was trying to figure out, how do I make the testing for this more rigorous, right? How do I show my other designer compatriots that this is a product that can really be seen on the market, and how science and R&D goes so well and is such a good match for product design and development later on. So, without ado, I will introduce Jonah to you now.
Bear with me, I have a lot of transitions, so I'm going to be clicking a lot, but it should hurt the presentation in any way. So, amazing introduction. I'm Jonah, and this is my project EcoSkin, and I'm stand by this statement. It is the next evolution in wound care. And, oh no, let's keep going. And what EcoSkin it's a fermented wound dressing made from Manuka honey and then fermented to a low pH to both accelerate wound healing and protect from existing bacteria infections. And I'll get into what it means, and I feel like an expert now being at Genspace, and it's very cool that I could actually say that. So this project means a lot to me. I have ulcerative colitis, and I have to be very careful with what I put on my body and how things affect my body. And I wanted to pick a project that is kind of been overlooked, particularly in the medical field, and I wanted to pick Band-Aids. Band-Aids by nature have been used for everybody. I'm sure everybody here has used a Band-Aid at some point in their life, but really been evolved in the last 100 years. Also, in the medical field, 90% of the tools that they use are single use, and they do that for sterility, but at the same time, 65% of the tools have PAFAS, which are forever chemicals, which again is a big issue when you're dealing with open wounds or very chronic wounds. Additionally, the rate of antibiotic resistance is increasing, and that might not seem like a big deal to you, and what was once very easy to treat, like chronic wounds or diabetic foot ulcers, is becoming more and more difficult, and that's because we're prescribing more and more antibiotics for very common things. I got my wisdom teeth out, and they gave me antibiotics as a preventative, even though I didn't have any antibiotics, even though I had no infection. So I want to focus on these 3 bacteria right here, because I'm going to go back and explain them later in the presentation. But these are bacteria kind of known for antibiotic resistance and also a lot in chronic wounds. And finally, in 164 years, dressings haven't really evolved. And they work well, and that's why they haven't evolved. But I was really wondering, why haven't they evolved? And can me as a product designer do better? So this goes into what's going on in the medical industry right now. So acetic acid, or also known as vinegar, is been used for thousands of years for wound healing, and they've actually found that a concentration of 1% is super efficacious at not only protecting against bacteria infections, but also accelerating wound healing. And they do this by lowering the wound, and actually what that does is increases the amount of fibroblast cells produced during the healing process, which actually in theory reduces scarring. So for C-sections or for face surgeries, they actually opt for acetic acid dressings. Also, Manuka honey, thank you for the lovely introduction, it was amazing. But Manuka honey is amazing, and it's used in the vet care settings because it's super antibacterial. Yes, it has hydrogen peroxide in it and it's low pH, but it's actually the formation of a crystal that actually creates a super antibacterial wound healing environment. And finally, bacterial cellulose. And what bacterial cellulose really is a kombucha SCOBY. It's just a really sterilized, perfectly hyper-processed way of creating cellulose. And it's been used because it's super water absorbent, so it creates a very moist wound environment. It's breathable, it's porous, it's really cheap to produce. And you can impregnate it with different medications. So people use honey, people use acetic acid. And the process of making it is the same way you make kombucha. I imagine everybody here has had kombucha at least once in their life. Maybe not the SCOBY on top, but the kombucha itself. But I was thinking, okay, if you take kombucha and it produces the cellulose fiber, well, what happens if you replace the sugar used to grow the SCOBY and you use a medical honey, and then you ferment it to a low pH, particularly a acid concentration of 1%? What would that do for the wound site? Would it accelerate wound healing? Would it do anything? And that was the introduction to my relationship with Genspace. And big thank you, because I could not have brought my product to a level of legitimacy or seriousness enough that Johnson & Johnson actually interested in partnering with me for this product, and it's all thanks to Genspace. So thank you, you guys are amazing. So I came to Genspace after seeing my own SCOBYs and really finding the optimal formulation. And I wanted to find something that was cost-effective, fast, easy to produce, and also could be mass manufactured. And so I came to Genspace with these bacterial sheets, and I test these. How do I prove that there's actually antibacterial properties with it? And that's something called the Kirby-Bauer diffusion process. And what it is it's a effective way, and a standardized method used in to measure how effective a substance is at stopping the rate of bacteria growth. And it's commonly used in hospitals, research labs, to test antibiotics, antiseptics, or antimicrobial materials. So the next time you use Neosporin, or the next time you take antibiotics, this is how they test it to prove that it works. And it's very, very easy. And it's very easy to do. All you do is you single out a type of bacteria strain that you're testing, you inoculate, suspend it in a nutrient broth, so you replicate it overnight, you take a little bit of solution, 25 microliters, you spread it on a type of petri dish called the Mueller-Hinton agar plate, and then you place your little antibiotic discs on it. And then after 24 hours, usually 24 hours of incubation, you measure the zone of inhibition, so essentially the radius between end to end that the bacteria creates is essentially that the rate of there's no bacteria growth, and you measure that. And that's exactly what I did. So I came in space, and remember those 3 bacteria that I wanted to test in the beginning that are super prone to antibacterial resistance? I tested those 3 at Genspace, and the result was pretty amazing. Not only did the SCOBY have antibacterial properties against all 3, but I actually tested it against common dressings on the market, particularly Neosporin, gauze, and hydrocolloid. And I'm not going to claim that it has stronger antibacterial properties because I do more testing about it, but the zone of inhibition for my SCOBYs were greater than the zone of inhibition shown for the Neosporin Band-Aids, which I think is a little bit of fermentation and a little bit of honey, you can achieve stronger antibacterial properties than something currently on the market. And I patented it because I'm like, holy shit, this is cool. I better myself. And because I'm a product designer, I thought, okay, this is amazing, this is great, but I want to turn this into a product. I want people to use this, both in hospital settings and in, conventional Band-Aid use settings. And what I wanted to make something that was comfortable, and I wanted to make something that was stable and also something that was familiar, something that people want to use. Not because it's in Band-Aids, but because they want to invest in their health, and they also want to invest in a future that's plastic-free. And so the first one was skin adhesion. So due to the nature of bacteriocellulose, it's super porous, and it's super, and it actually could absorb a lot of water. And I wanted to create something that was naturally sticky. So it is naturally sticky because of the honey and the cellulose. But the actual texture, I wanted it to be pretty minimally invasive. So when you put on your clothing, it doesn't, or when you touch it, you don't have a sticky residue on it. And it all came down to how I dehydrated it. And so I'm giving you a little bit of IP secrets right now, but I found that a temperature of 45 or 12 to 16 hours produced a cellulose fiber that was really sticky, but not overly sticky. Just enough sticky that it actually sticked on your hand, but not too sticky where you don't have sticky hands, because I hate sticky hands. Also, with the packaging, I wanted to be sustainable. How many times have you had to open a band, you can never find the tab, and you rip it off, and then you have the paper backing, and then the plastic backing on the inside? You have too many materials just to get to the Band-Aid. So, because I am a fermentation researcher and my background is in food and food fermentation, I thought it would be really beautiful if we could package them in vacuum bags. Both because it can actually, you're taking away the oxygen, you're creating a shelf-stable product, but at the same time, really beautiful. It's very poetic. You could actually see inside the bag. You see what the contents are. And that's kind of what this whole project is about. It's about transparency, and it's about really highlighting the natural properties of each ingredient. But it annoyed me that I was still using BPA bags until I found cellulose bags. So the bags that I package EcoSkin now is fully biodegradable. So the entire packaging is biodegradable. Even the ink that I print the packaging on is biodegradable. And you can see some testing there. And as far as size and shape, I wanted to create shapes that are kind of familiar. It's a little bit of an unconventional product, and I want somebody to put this on their skin and use this as a wound healing, so I wanted to come up with shapes that are both familiar and not super out of the ordinary. So I kind of have a big one and the typical Band-Aid one, and that's it. And then essentially just some lifestyle images. This is one called the EcoSkin Strip, so it wraps around your finger. This is called the EcoSkin patch, and it could be anywhere on your forearm, on your neck, kind of wherever you want. And then this is the EcoSkin wrap, where it's the really big one. So if you have a big wound, or if you have something that kind of needs to be covered. And it also works on non-hairy legs. I had to test this because that was a big what if, you know, could it actually... This is Mitch, he's one of my best friends. He's got the hairiest legs that I've ever seen. And they stuck extremely well to it. And I'm gonna leave my presentation with how you actually apply it and how it works. So, like I said, with internal packaging, I want it to be easy. So there are proliferation tabs along the perimeter of the Band-Aid. And then, like you do with any sterile hospital dressing, you peel. And then you throw in the compost, because it's compostable. You take the Band-Aid, carefully peeling it off. You wrap it around your finger or anywhere else that you have a wound. You give it a small squeeze, and you're good to go. You're investing in the future of healing, and that's my project. Any questions? Yeah, what's up? Sorry.
If you have like a wound that just like will not quit bleeding,
Mm-hmm.
how does like contact with blood, I guess, affect?
Question. So bacteriocellulose in nature is super porous, and it actually can absorb up to 700 times its own weight in water, liquid, or blood. So the only testing that I can really do ethically is on my friends and family. So whenever I get a cut, I do a little. I don't do it on purpose. I do totally like, you know, I promise. It's totally accidental, and I'm like, oh, you know, why not? Like, it always use a little testing. But it actually absorbs really well. So similar to how hydrocolloid actually absorbs any material, it actually will absorb the blood into the wound sheet itself. So it won't get under clothing, and it won't actually leak out. It'll just absorb into the Band-Aid, so it'll puff up a little bit.
Let's go. Love it.
Yeah.
So when you did the testing,
Uh-huh.
is the material, I guess, because sometimes when computer study is alive, and like simply having something living will deter the other bacteria from contaminating. So like, is your Scoby Band-Aid like dead, or is it like?
It's alive, and that's a really, really great question. So further testing that I could do is actually determine what is it about the SCOBY and the nature of the SCOBY is aiding in the antibacterial properties. Is it the honey? Is it the acetic acid? Is it the low pH? Is it the bacteria already present, responsible for the cellulose growth? Or is there something kind of magical and synergistic happening during the fermentation process that creates a new antibiotic by itself? So I'm actually I just made my Petri dishes last week, and that's further time that I'm going to do before Biodesign Challenge.
But at its current state, would it be like food and have a shelf life? Like, because you're doing the food wrapping to prevent the oxidation, because it's like food. It's almost like
So
food wrapping.
you say that. So the way that I'm combating that is I'm dehydrating it, and I'm dehydrating it at a temperature that doesn't kill the bacteria, but it makes them dormant until it comes into contact with your skin that rehydrates it. So your contact with the skin actually redormatizes the bacteria, hence aiding in the antibacterial properties. And because it's actually an oxygenless environment, kombucha is an anaerobic process. If there's no oxygen, it's essentially dormant. It's the same way that probiotic pills you freeze-dried until they kind of interact with your microbiome. That's how I kind of modeled my activation.
Very cool.
Thank you. Yeah.
So I have one question and one suggestion.
Sure.
So one question is, how long does it take to be biodegradable, the old product?
So that's a really good question. I haven't done any formal testing, but cellulose by the nature, a couple of studies that I found that it could decompose in a matter of a couple of months, which is pretty cool.
And one suggestion is,
Sure.
I can see you really like minimal design, and you hold like you prefer more minimal thing. Like if there is more wide range of sea color, that might be really good.
You know, it's funny that you mentioned that. It's semi-transparent, so the way around it, it actually reflects the color of skin that you have. So I have naturally lighter skin, so it actually is a lighter on me. But if you'd actually notice, it's actually darker on the mannequin hand. So that's actually the natural color on me, but it is semi-transparent, so it does reflect your skin tone naturally. Additionally, because it's semi-transparent, it actually, you could see the wound site in real time. So for surgeons that want to monitor a wound site without actually peeling the, you could actually monitor through the SCOBY and see, is it healing properly, not healing properly, do we need to intervene, do we not need to intervene? Yeah.
How long does each Band-Aid last? How often do you need to replace it?
So that's a good question. The longest that I've used a Band-Aid for is 24 hours. But it's funny, my dad has a prosthetic leg, and he uses them actually because there's a lot of chafing where the socket meets the new leg, and he keeps them on for 48 hours. And so he says they're like a medicated moleskin, where the more your skin sweats, the more it kind of puffs up. And also, he says there's continued, he's a doctor, so, everything with a grain of salt, and, he won't sue me because I'm his son, so if, his leg is infected, no harm, no foul. But what he says is, he said, actually, he came to New York recently, and he walked around Central Park, and he had a huge kind of gap around his leg, and he said in a matter of days, there's a new skin actually developing underneath Toby. So there's a whole slew of untapped potential that I need to research a little more. More so, I need to partner up with a larger organization that actually legitimate science, because my bread and butter is industrial design. I can make things look really beautiful, but legitimate science, I think that's going to take a little more of a professional institution that I hope to partner with soon.
Say we can have more questions for Jonah. As we go on, we have one more speaker, okay?
Thank you, thank you.
So our last speaker for the evening is Marin. She is an artist here, a bioartist, who is working in bioplastics. She has a wonderful formulation of pieces that have been on display recently. And so I can't wait for you to see her beautiful work and see how biomaterials, how you can go from learning these things in school and really creating and forging a career forward for yourself and showing people what future with less single-use plastics can be incredibly beautiful. And so without further ado, Marin.
It's nice to see everyone. I don't know if you need to stretch. I know in a long session, please feel free to get up or move or whatever your body needs. I will try to be quick. I do have quite a few slides, so I'm just gonna go quickly through them and then we can talk about it. I think the project that I'm presenting today is I really want you to sort of shift your perspective from a lot of what we've been listening to. I use science, but not for a sort of a productive end, and I'm really interested in the poetics and possibilities of materials and sort of those moments between life and death that we experience with and alongside other species as other plants within our ecosystem. My name is Supa Marin. I'm currently on the Silva Arts residency at the World Trade Center, where I have a studio this year. It's the luxury of working in this really picturesque environment. I've been working with Genspace, I think since 2020 now, and it's been sort of the most incredible partnership of my life. I was born in India and I grew up in France, and then I went back to India, and sort of hybridity, different things were really part of my upbringing, different cultures, different ways of being. I practiced architecture for a decade in New Delhi, in this moment in India where we were rapid urbanization, and I really got to witness how we as architects worked with and on land, often in pretty destructive ways. About a decade after working is how I moved closer to an artistic practice, and a lot of work now today is a rethinking of architecture, but also a critique, also using its methodologies maybe against itself in some manner. I'm really interested in various forms of vernacular architecture in India and in alternative materials that I've been in over the last few years. I'm going to share with you today some practices, what I call practices and para-practices within my work. And I really do believe in process, and I really do believe that it is the para-practices, the foundation, the relationships, the learning, the ideation, the development that creates a new idea, a new vision. So 2022-23, oops. This is a site-specific sculpture that I did in 23 in Switzerland, a residency. It was a 2-month residency in a sort of a winemaking facility in a vineyard in this beautiful picturesque landscape in the French part of Switzerland. All of the trees were sourced from the site, all of the materials of the sculpture that you're witnessing. So it's a pre-existing architecture, and my work is what is sort of developed site-specifically within that space. This piece is called Feel. I often have pretty long titles that are sort of snippet from my journal entries as I travel that are part of the work or sort of adjacent to the work. I'm going to read this one out to you. It's called Field Maple. At the end of the world, dark thunder rolls to the footsteps of men chasing uninvited, scavenging crows. The nostalgic scent of wet, dry earth mixes urine, dung, and sand. And that's about 15 feet tall, 2 fallen maple trees, bioplastic. At the same residency, the second sculpture that I developed from this bioplastic, that I will get into the material and the background. This piece is called Feel Linden. I am the prism to your shadows. Through my eyes, pinks turn to crimson, grays to silver, yellows to gold. As I roam the midst of your strange lands, flow like veins across my open sky. I'm interested in witnessing otherness as myself, and I'm often not at home. I'm often out of my comfort zone or within a different context, and I'm interested in that encounter with a so-called other. That is me, that is you. And often my work points to these encounters, the sculpture turning towards the human, the sort of the human, non-human, meeting each other. A third piece, as you can see, another site-specific rendition. In this case, there's 2 birch trees interwoven. I often build sort of like complex tensile structures with fallen limbs, and I bend them, I curve them, and I build these sort of like become really strong meshes from the tree structures, over which I can do pretty complicated mold making processes with my. And then one of the most exciting things is that I can draw on top of the molds, I can use the material and layer ad infinitum over itself, and that's sort of the aesthetics that I'm interested in. I'm gonna keep moving on, but you get a sense of the work. A week before I arrived at the residency, which happened to be in the town where Corbusier was born, a freak tornado sort of that was like really not normal for the area, destroyed all of the trees in the city. So over 150,000 trees were torn down. And what I thought in my residency was like completely different, because I arrived in this landscape and everything was on the ground. This was like the that I witnessed. And so a lot of the work really became about like talking to people, understanding the history of space, and really thinking about these plants as sort of memorials, events that are transpiring globally at a higher rate. And all of the grass that I used was from the manicured lawns of the landscape of the That's sort of the process of making my sculptures. I often do a lot of site studies and background research in a context where I work site specifically. And the way that I approach this project, I'm sort of backtracking now, so you have a sense of what the work looks like today. But how did I get there? I was working on a public art project in the city of Oakland. This is Oakland City, and this is its public plaza, which is essentially a giant lawn. And I got access to all of the brief documents that the city had developed for the redesign of this extremely modernist kind of panoptic space. And through accessing these documents, I sort of really understood why my body was so uncomfortable in this space. And my proposal to the city was just to pause the mowing of the lawn for one season. I was interested to see what could emerge in land that was already ravaged or erased or homogenized. And I was really thinking about, specifically in the US, the mowing of the lawn as a constant repetition of erasure, of history, of diversity, of difference. Weirdly, they agreed. I don't know how, but I think that there was this like deep fear and fascination of what would happen if we went wild. But at this point, I was so attached to those grass clippings because at the end of the season, I had all of these bits of dead grass, of grass compost. And my question became, can I make a viable material for sculpture for my practice with this material? And that's how I was introduced to Genspace. We're now in 2020 in the deep pandemic, where I actually made the most amazing friends I've ever had inside of the in Brooklyn. And Jill, who was actually at the biomaterials lab here at Genspace, met me for the first time ever at a taqueria in Bushwick, and we spent 1 year masked in her studio developing these materials together without knowing each other before, without even knowing what each other looked like. And over time, so many people joined in on the project. So we started collecting. I had a hookup with Joseph, who maintains the landscapes at Greenwood Cemetery. And I would call him and be like, I'm coming. And I would go hang out with the Moors, and I would like fill trash bags full of grass and mowing. A lot of interesting sort of side conversation around sort of the care of lawns and the maintenance, and also the workers who do that labor. And then we would bring in, we tried hundreds of different recipes until we had a range of successful materials. I'm really at this point starting to think about the act of labor of making my own material autonomy as a sort of an act of resistance, and I'm really thinking about that in relation to various civil disobedience that are the history of India in relation to craft and manufacturing and small-scale industry. So what you see on this side is grass, pure grass clippings, when it's more paper-like. And what you see on that end is when it's more plasticized. And so this entire range of visual palettes is what we were able to develop. And based on various properties that I need in my work, I can sort of make different combinations. These recipes, we also did 3D printing with the material. We were on a residency at the robotics lab at the Brooklyn Navy Yard. We, in all of 2021, had our first research exhibition where we showed all of the prototypes and samples. We tested how they age and how they decompose and destroy outside, and I'm really interested in sculpture as this growing and dying process rather than just a market-driven investment. And I'll end by saying that last year I had the opportunity to show my work in response to the sculptor Auguste Rodin, and I'm interested in the relationship between these hefty bronze historical works and language of plants and grasses. I'm also really interested in sort of the vernacular history of grass in Asia and Africa. So what you're seeing here are the first currencies that were sort of woven grass mats that were exchanged as the first version of money in these these are from the Congo, and these were part of the museum's collection that I could use. That's all I have today. Thank you so much. Any questions?
In the beginning, we were talking about Western and, well, East philosophies, I guess, in a way. And there's, I think that you were mentioning something about how it's like maintaining the wisdom such from older generations rather than just brushing it off. And I'm wondering if you see any connections between Eastern and Western archer and how it's developed and those differences in philosophies.
I mean, that's such a big, vast question. I am really interested. I don't know if you're familiar with cow dung as a construction material, but in India, in rural India, for example, we still use it. You collect the cow dung and then you put it out to dry, and when it dries, it sort of becomes this like very strong, fibrous thing, and it's used for natural gas as well as building construction. And I think a lot of the lineages that I'm sort of mapping and tracing, I'm not using the cow dung directly, but I'm using the source material that the cow is eating, and I'm interested in that process. And so I think in a way I'm talking about the past, but also in a way I am talking about future, material science as this sort of like fetishized technologic process that we see before us, how it's in Silicon Valley and beyond. But I'm more interested in like, what does that mean as something more embodied, maybe not as sexy as silver looks or like as your cool robots are these days, but a sort of like shadow self maybe.
Wait, you might have touched on this. What happened with Oakland? I mean, like, what happened after the year? Did they grow back? Like, was it kind of wild?
Yeah, it rewilded, and it isn't, because it's all Bermuda grass, which, I mean, I knew this going into it, which is part of the interesting thing, is Bermuda grass is genetically modified so that we replant it every year, because it's all patented, and it's not allowed to go to seed. And so it's really designed, it's like a very highly designed material, so it's never going to grow more than, but the sort of like the wildflowers came in, sort of like strange ecologies, the rewilding. It was weird because it happened just as the pandemic hit as well. And so it sort of was the sync up with the world where everyone went on pause. And so it didn't get any pushback because it seemed so natural. All right, so much.
All right, lovely humans. So if you are impressed with the things here, or you are feeling inspired, want to do some work yourself, there are opportunities to join the lab. We have 3 different tiers of membership. If you want to just get started, meet some community people who are in our community, and you want to join one of our 3 community projects. They include Biomaterials Studio, so if you're in any way interested in mycelium, bioplastic, biomaterials, anything in that realm, you can definitely chat with them. They will be sort of housed near the biomaterial wall. Another one of our fabulous community projects is Open Plant, which since the last Open Lab has had some very exciting developments. They have been able to transform their mercantia to include the insulin gene. And now they are trying to understand the expression of that gene in the Mercantia plants. And so if you have any questions about that wonderful project, Dave and Tina and Nitin in the back are also here. They have wonderful Mercantia plants over there on that side of the lab or here as well. So you can reach them. We also have another group called Microbial Reef. They have been working on sequencing the microbes in the water near an oyster reef, sort of the bay near us here in Sunset Park. We're able to publish some of their genome data. And so if you want to chat with them about their data as well, we have a few folks from Microbial Reef here too. Around here, they're also in the process of building a Raman spectrophotometer. And so a DIY Raman spectrophotometer. So that's a way that you can understand the genomes of certain microbes as well with a longer read than our beautiful, gorgeous mini sequel right here. And so, if you are interested in joining these lovely folks in their endeavors, you can find the date and times of those projects on the whiteboard. If you have ideas and experiments you want to do yourself, individual membership might be for you. You are able to come into the lab 24/7, and you have storage here and are able to have access to our wonderful community of mentors, scientists, designers to sort of hopefully found the new Opentrons. Aha. And then if you are yourself in the throes of trying to build a business and you're not ready yet for an incubator and you want to test out a couple more ideas, premium membership might be for you. You get even more storage in the lab and more access to different kinds of equipment and such. If any of these things interest you, feel free to chat with me or anybody on staff. We'd love to have you here in the lab. I will also note that we have open nights and public programs as well. And so if this is something you're really interested in, all of the event happenings here, definitely check out our newsletter, which is going to be the community project meetings. And then this is the QR code, if anyone's interested. But we also do offer classes and workshops as well. So let's say you're not ready to join worship, you are more than welcome to join some of our classes. Some of them that are coming up in June include an intro to plant tissue culture. Wow! One of our community project leads, Jazz Neal, who's wonderful. Then we also have synthetic biology and biohacker boot camp, 2 classes that are lab intensives that Casey had mentioned at the beginning of the presentation. We are introducing a protein hacker boot camp. So if you are interested in learning how to isolate proteins and run different kinds of gels, we also are offering that at the end of the month, end of July. So please check out all this on our website. But shall I? Okay, but if you want to find out anything and more about the wonderful things we do here, please subscribe to our newsletter and also donate. We're a small but mighty nonprofit doing amazingly wonderful things. And so if you have it in your hearts, please donate to us. We are also having a wonderful birthday bash in the fall. And so if you are in any way looking to get involved, you want to join us as a mentor, as a volunteer, as someone to help get the word out about the fabulous things we do here, please chat with us, sign up for that newsletter, and also donate. Donate. So, with that, we want to thank the FIT program and Dr. Karen Pearson for all of the wonderful things. Oh, do you want? Oh, I thought you wanted me to do it. Please do the thing. You're our sponsor. Everyone, don't worry.
I have to sit because I'm heavy and so are my ankles. You can see me still. Hello everyone, I'm Vanessa. I'm the Development and Communications Manager here at Genspace. Please also follow us on Instagram, we're on TikTok, we're on LinkedIn, and we are streaming on YouTube for the very first time, so that's really exciting. Thank you for being here, and we hope so many new faces. I hope a new friend tonight, or 2 new friends, and I hope you come back for our programming and just to hang out or join a community project. I want to do a special thank you to FIT Genspace Scholars. It's always a treat to have you. And like Serena mentioned, birthday bash, sneak peek date is September 4th. So if you are very soon, you'll be getting some information about our save the date and information about attending the event. It's a great party. And so please sign up for the newsletter so that you can get the information. And And if you're really into event planning and would like to assist in putting together the birthday bash, please feel free to reach out to me. I will be here for the remaining of the evening. Want to give you some time now after this to be able to get back to mixing and mingling for the evening. I just want to be sure to, there are so many people that help us put Open Lab together, and we're just so grateful. So I want to take this time to give some shout out to everyone that helped us make this a magical evening. It's so wonderful to have a small and mighty staff. So it's really, really implemental to have volunteers that assist us. So thank you from the bottom of our hearts. Thank you, Serena, for always leading this wonderful event. And also, you must clap for Serena's suit. Please stand. Thank you to our lab stewardship interns who have been for the past few months helping us. On Fridays, they help us get the lab ready for classes and workshop and just get ready for the week. Nico, Sarah, Izzy, who's in the back here, for always being so helpful in organizing the lab. Our photographer, Sarai, and our volunteers who helped us set up, John, Nelson, Anson, Carmi, Adasa, Diana, Anna, I miss anyone, but thank you so much for helping us pull it together in record timing. I feel like today went very smoothly. Thank you for that. And finally, we could not do this work without our generous sponsors and support for our donors. He's donated for us to get some snacks for this evening. Thank you for that. And also, especially thank you for Government, University Foundation, and corporate partners, the teams at Con Edison, Pinkerton Foundation, New York State Council of the Arts, and the Experiment.com Foundation. Thank you to FIT and the Office of Academic Affairs and the Office of the President for funding the FIT Scholars. It's been a pleasure to have seen, I've been here since 2021, so I've seen a few cohorts of the FIT Scholars Program, and it's just so interesting. There's just like so different in their projects every year, so it's like a really special thing to have them here, so thank you. So I also, a little bit of cleanup, so just so that back to mingling, if you don't mind helping us arrange the chairs a little bit so we can have some more space for us to get around, that'd be great. And yeah, enjoy your evening. Thank you. Look at my ankles, bro. Yeah!
I think just stacking it and then putting it in some location, someplace, somewhere. But stacking is probably the first thing we can That's much better. So there's something that is the week, but that's
people